Hello everyone, I am Sivani Murikesan, studying first MSc Microbiology at Vivekananda Arts and Science College for Women, Sangeri, Salem. This presentation is my part of my coursework in the subject Bioremediation undertaken by Dr. R. Denis Mercer, Assistant Professor, Department of Microbiology, Viewers. Let us discuss about an important topic in microbial leaching of ores and its process. In this video, we are going to learning about what is bioleaching, microorganisms and mechanisms involved in bioleaching, types, factors affecting advantages and disadvantages of bioleaching. First, we discuss about what is bioleaching. Bioleaching is a biotechnological process used to extract the metals from the ores with the help of living microorganisms. This process uses naturally occurring bacteria to recover valuable metals in the eco-friendly manner. This process, also known as microbial leaching or biomining, it mainly involves interaction between microorganisms and minerals, where microbes convert insoluble metals components to soluble forms. Bioleaching is especially useful for extraction metals from low-grade ores, which containing only small percentage of valuable metals. Traditionally, extraction methods are expensive and inefficient for each use, but bioleaching makes the process economically favorable. The modern era of biomining began after the recovery of bacterium Thiobacillus peroxidans, which is known as also Acidothiobacillus peroxidans. This bacterium plays a key role of bioleaching because it can oxidize the iron and sulfur components. During the oxidation process, metals like copper, gold, uranium, nickel are released for their use. On the right side of the picture, we can see the microscopic image of bacterium involved in bioleaching. Thiobacillus species, Leptospirillum species, and thermophilic bacteria are mainly involved in bioleaching process. The important group of bioleaching process is Thiobacillus species, which are under the genus of Acidothiobacillus. These bacteria are acidophilic, autotrophic, and capable of oxidizing iron and sulfur components. Some important species are Thiobacillus thioxidans, Thiobacillus peroxidans, and Thiobacillus porophorus and Thiobacillus cuprinus. The Thiobacillus thioxidans is the bacterium oxidized sulfur into sulfuric acid. This acid produced helps dissolve the metal sulfides, make the metal soluble. Thiobacillus peroxidans is the important bacterium in bioleaching. It oxidizes ferrous ion into ferric ion. Ferric ion in the chemically attracts metal sulfide ores, release metal like copper and uranium. Thiobacillus porophorus, the species highly halotolerant, meaning it can survive in saline environment and useful for bioleaching in salinity condition. Thiobacillus cuprinus, the bacterium involved the copper bioleaching and tolerant in high copper concentration. Leptospirillum species, especially Leptospirillum peroxidans, this bacterium strictly oxidizes ferrous ion, plays a major role in maintaining high ferric ion concentration, enhance the oxidation of sulfide minerals. It highly efficient in ion oxidation and commonly found in acidic main drainage environment. Leptospirillum species, especially Leptospirillum peroxidans, this bacterium strictly oxidizes ferrous ion, plays a major role in maintaining high ferric ion concentration, enhance the oxidation of sulfide minerals. It highly efficient in ion oxidation and commonly found in acidic main drainage environment. Thiobacillus bacteria, which grow at high temperatures. These are especially important in high temperature bioleaching operation. For important example species are Acinus berylli and Sulfobacillus. Acinus berylli, it can oxidize both sulfur and ion at high temperature, and it is used to industrial bioleaching refractory ions. Sulfobacillus is the thermosulfooxidants. It is highly highly thermophilic bacterium that oxides sulfur and ion components and improve metal recovery efficiency at lower temperature. There are two mechanisms are mainly involved in bioleaching. The mechanisms are direct bioleaching and indirect bioleaching. The first mechanism is direct bioleaching. Direct bioleaching is also known as contact bioleaching. Direct bioleaching occurs in the physical contact between the microorganisms and the minerals on the surface of pores. The bacteria which attach themselves to minerals and form the thin layer is biofilm. The microorganisms carry out the enzymatic oxidation reactions directly on the mineral surface. The oxidation takes the place the surface of series of the biological oxidation steps. Bacteria are attached to the sulfide material to like pyrite. The microorganisms mainly oxidize ferrous ion to ferric ion, sulfur components to sulfide. For example, oxidation of pyrites. The common example is oxidation of pyrite. Pyrites react with oxygen and water in the form of ferrous sulfate and sulfuric acid. The production of sulfuric acid helps maintain the acidic environment, which further enhance the metal. Indirect bioleaching. Indirect bioleaching is the mechanism the microorganisms do not directly attach to the mineral surface. They produce strong oxidizing agent which chemically react with the metal ores and help in the metal extraction. Microorganisms oxidize the ferrous ion into ferric ion. They also oxidize sulfur compounds to sulfuric compounds. They produce sulfuric ion and ferric ion. They are powerfully oxidizing agent. Ferric ion then chemically react with the metal sulfides present in the ores and dissolve in the metal into the solution. For example, bioleaching, the ferric sulfate act as the oxidizing agent. Insoluble uranium is covered into the soluble uranium sulfate. Ferric ion is reduced back to the ferrous ion. The bioleaching process is the three type of bioleaching: slope bioleaching or dump bioleaching, hill bioleaching, and in-shed bioleaching. First, we discuss about the slope bioleaching or dump bioleaching. Slope bioleaching is the process the ores finely grounded and placed on the large piles on the slope surface. An aqueous solution containing microorganisms is continuously sprinkled over the ores. It was the water containing bacteria such as thiobacillus which help the oxidizing in the metal and releasing the metal in the solution. As the solution flows the grounded along with the slope, it dissolves the mineral from the ores. The mineral rich solution is collected in the bottom of the slope. The collected solution is sent in the oxidation bond and where the metals is extracted and the bacteria are regenerated for reuse. Heap bioleaching. In heap bioleaching, ores the arranged in heap and goes through the same procedure as the slope bioleaching. The ores is dumped into the large heap is called leaching heaps. Water containing inoculum of thiobacillus is continuously sprinkled over the ores. Water is collected from the bottom and used to extract the metal and regenerate bacteria as an oxidizing bond. In-shed bioleaching is the last type of bioleaching. This method extracted from the metal ores are not removed from the earth. The remaining ores is original procedure underground. First, surface blasting or drilling is carried out by the increasing of permanently rock so that water is passed through the easily. The water containing bacteria such as thiobacillus is pumped through the drilling pages into the ores body. The oxidizing solution is react with the ores and dissolve the metal. The metal-rich solution slowly dips down through the rock and it is collected from the bottom. The solution is pumped out and extract the metal. After extraction, the solution containing bacteria is reused for their leaching, making the process is economical and efficient. Next, we discuss about the process of bioleaching. What are the processes are involved in the bioleaching process? Preparation of ores, inoculation of the microorganisms, microbial oxidation of iron and sulfur, regeneration cycle, formation of the leaching liquids, and recovery of the metal. The first process is preparation of the ores. First, crushed and grounded in the small particles of the ores increase the surface area and allowing the better microbial react or attachment. Fine particles are improved between the microbes, oxygen, water, and mineral. The second process is inoculation of the microorganisms. There are two microorganisms are mainly involved in the process. The organisms is Thiobacillus peroxidans and Thiobacillus thioxidans. The organisms are mainly in acidic environment. The pH is 1.5 to 3. They are attached to surface of the metal and sulfide minerals and form the biofilm. The third process is microbial oxidation of iron and sulfur. Oxidation of ferrous ion is the microorganisms oxidize the ferrous ion into ferric ion. The powerful oxidizing agent. Sulfur components are converted into sulfuric acid. The pH is maintained was very important. The fourth process is regeneration cycle. Is the process is cyclic in nature, converted into soluble ion form. Ferrous ion regenerated and bacteria are reused. The solution is recycled back the heap and the type of the leaching. The next process is formation of the leaching liquids. The solvate metal ion accumulated in the solution. The solution is also called pregnant leaching solution. The solution is contain metal and sulfuric acid. The high rich metals is present in the leaching solution. The last process is recovery of the metal. The dissolved and extract the metal is recovery in the method of electrowinning, chemical precipitation, and solvents extraction. What are the factors are affecting in the bioleaching process? We choose the bacteria, pH and temperature, pollution density, metal tolerance, oxidation reaction composition and activity, and surface area. These factors are mainly affected in the bioleaching process. Application of the bioleaching. Bioleaching is applied in environmental solubilization, emission reduction, cost-effective extraction, and optimize the recovery process. What are the benefits of bioleaching is simple process. It is inexpensive technique. No poisonous sulfur dioxide emission in smelter. No needed for high temperature and high pressure. Ideal for low-grade sulfide ores, environmental eco-friendly. Advantages of bioleaching is it is a simple process, eco-friendly, employed for collecting the metal from waste and drainage, used to extract the refined and expensive metal, which is not possible by the other chemical process. There is no need for high temperature and high pressure, low-grade sulfide ores, and no poisonous in sulfur dioxide emission in the smelter. The main disadvantages is it is a time consuming process. It takes the 6 to 12 months duration or long time duration. It has a very low yield of metals. Require in large open area to treatment. It may be no process control. At a high risk of contamination. Limitation of the bioleaching. Time consuming, low metal yield, space requirement, process control changes. This picture is the overview of bioleaching process and extraction of the bioleaching. Thank you for watching this video.